So let's take a minute and review the enzymes lab that we did this semester. So first thing we want to recall is that an enzyme is a protein, okay? And remember that proteins are made up of amino acids. And as we'll talk about a little bit later in our review for this lab exam, every protein is essentially instructed for by a gene, right? So a gene sequence gets transcribed and translated into a protein, and an enzyme is a specific type of protein. We have structural proteins like collagen, other kinds of proteins, but an enzyme is one type of protein. Now, as you see shown in purple, okay, this is our example enzyme, and I want to remind you of a few things with enzymes before we get into our actual lab activities that we did. And the first is that an enzyme, it is typically very specific to a particular substrate. So it's kind of like a lock and key. The key that you use to unlock your house is different than the key that you use for your car, and that is the way enzymes and substrates typically are. They have a specific recognition for one another. Now, where the enzyme and substrate interact is called the active site. And as you can see, the enzyme helps what we call catalyze a reaction. It really lowers the energy of activation or activation energy, and I'm going to talk a little bit more about that. But one way that it does that, as you'll see, is in this case we have a substrate that is actually going to be broken up into two different products. But sometimes we have two substrates that a bond is produced between the two to create a single product. So it brings these substrates or reactants into close proximity to one another so that the reaction can move forward. Now, I mentioned energy of activation or activation energy. So if we look at this graph and we say that this is sort of the time progressing this way for a chemical reaction, and on the y-axis we have the amount of energy increasing, okay? Let's say that we have a certain amount of energy right here in our substrate, and or we could say in our reactants in the beginning molecules. And at the end, in our products, we have a certain amount of energy, so we have much less energy in our products. Okay, you can recall that this is called an exergonic reaction because energy should be released. But we know that it doesn't directly go from that amount of energy down. That's not the way it actually works. In reality, in order to get the reaction going, there's sort of this large hump that has to be overcome. And that amount of energy from where you're starting to the hump here, this amount of energy that has to be put in by the cell is called the energy of activation. Now, what an enzyme does, so let's use a different color. An enzyme comes along and it lowers, okay, that hump, or the energy of activation, the amount of energy that the cell has to put forward to get this reaction to move. Now, let's spend a little time talking about your specific activities that you did for your enzyme lab. But a couple other things before we move on that I want to mention after I clear this off is that enzymes can actually be affected by inhibitors, okay? And there can be competitive and non-competitive inhibitors. If an inhibitor is competitive, that means that it is going to compete with the substrate for the active site. So it's going to bind to the enzyme in the exact same spot as the substrate. Okay? If it's non-competitive, okay, then it's going to bind somewhere else on the enzyme. For example, it may bind over here on this side, but by doing that, it will change the shape of the active site so that the substrate can no longer bind to the active site. So inhibitors come in two different types, competitive and non-competitive, but both inhibit the enzyme's ability to bind to the substrate. Now, another couple of things is that all enzymes have optimal pHs and optimal temperatures that they work the best. They may function outside of that range, but they have an optimum pH and temperature that they work the best. They also have temperatures and pHs that can do something called denature the enzymes, okay? And denature, remember that enzymes are not alive, right? They're not living cells. They're smaller than a cell, so they're not living. And so we can't say that they're killed, but denature means that they're no longer functioning. They're no longer to carry out the catalysis of whatever reaction that they're responsible for. So let's talk about our first experiment. Okay, so experiment 1 is dealing with enzymes in food. And our main focus here is an enzyme called amylase. And you have this enzyme in your saliva because the job of amylase is to begin to break down starch. Now, what is starch? Starch is a very large polysaccharide. It's many glucose sugars bonded together. So what amylase does is it begins to break the starch down into its individual glucose molecules. Okay, which glucose is a monosaccharide, right? A single sugar. So that's the job, that's the reaction that amylase carries out in the cell. One thing I want to point out to you, notice that amylase ends in ASE. Many enzymes have that ending. Polymerase, catalase, amylase. That's a typical ending for the name of an enzyme. All right, the other important thing about amylase, besides being found in your saliva, amylase is also found in many vegetables or fruits, okay? Naturally occurring there. And so in this particular experiment, so this quadrant represents a paper towel, and it's divided into 4 quadrants. Remember that you sprayed a starch solution all over the entire paper towel and allowed it to dry. Now, I want to remind you something called IKI, and this stands for iodine, potassium iodide. The reason that the K means potassium, so that's why it's IKI. I will probably just refer to this as iodine as we're going through. What is this? It's an indicator for the presence of starch. Because what happens when iodine or IKI and starch meet, you get this really dark purpley blue black color that develops. Now, normally, when iodine is not in the presence of starch, it's yellow. That's the natural color of it. But when it reacts with starch, it produces this purple-black color. So in this experiment, you used iodine or IKI to determine whether or not starch was present. So in our quadrants, this was supposed to be the positive control. This was supposed to be the negative control. And these two were foods of your choice. Okay, you got to choose what you wanted to put in these quadrants. So it was recommended to use ginger root for this particular quadrant. I believe also banana was recommended if no ginger root was found. And you were to choose an appropriate negative control. So I would say one of the best choices would be just put a little water there, right? We know that water does not contain any amylase. And we're looking to determine, was there any amylase in any of these food sources? Okay, so remember the paper towel, you applied the starch first, allowed it to dry. Then you took your appropriate food and you rubbed the food onto the paper towel so that if there was amylase in the cells of that food, it would transfer to the paper towel. Well, the amylase then would go about breaking down the starch into glucose. Glucose does not react with iodine. Okay? Therefore, if starch was present, you should get a purple-black color. If starch wasn't present, you get yellow. So what you should have seen, this quadrant should have been sort of a yellowy orange color because the ginger root contained amylase. The amylase broke down the starch that you had put on the paper towel, and therefore there was no starch to react with the iodine when you applied it. In this quadrant, you should have seen the purple-black color because we didn't apply anything with amylase. So the starch was present. Once the IKI was added, the reaction would have occurred, right, to get purple-black. Now, depending on what you used for your food sources, you may have gotten yellow or black, depending on whether or not they contained any amylase in them. Okay, now we're going to move on to the next experiment that you performed in the enzyme lab. Okay, this one, experiment 2, and we were interested in temperature's effect on enzyme activity. And remember, I mentioned to you that all enzymes have an optimal temperature. They also have a certain range in which they are effective. And the enzyme in this particular experiment was catalase. Notice the ASE. Now it's important for us to know, what does catalase do? Well, it catalyzes this reaction of hydrogen peroxide into water and oxygen gas. And what you were measuring in this particular activity is the amount of gas present. Okay, so what you said was the more that this enzyme occurs, right, the more that catalase catalyzes this reaction, the more oxygen gas will be produced, and that's what you were measuring, is how much gas was actually produced. Now, how did you do that? Well, you had a setup with your test tubes, and in the test tube, if you'll recall, you in each test tube you had multiple test tubes. You had test tube 1, test tube 2, test tube 3. Each test tube got the same amount of hydrogen peroxide, okay, which is the substrate, right, for the enzyme. And what was different was the temperature. So on the temperatures, you had this one, number 1 was in the fridge, so somewhere around maybe 10 degrees Celsius. This one, or 5 degrees Celsius, this one was at room temperature, okay, 24 or so degrees Celsius. And this one was not quite boiling. You let it cool a little bit after you boiled it, but about 85 degrees Celsius. So we had a range in temperature. And then what you did is you used yeast, okay? What was the point of the yeast? Yeast supplied the catalase. In other words, the yeast cells have the catalase enzyme. So the yeast is what provided the catalase for this reaction. So the yeast you put in the little balloon so that when you had the balloon fastened on top of the tube, you could dump the yeast into the tube, right? And that would allow the catalase to have contact with the hydrogen peroxide. So all 3 tubes had the substrate, which was hydrogen peroxide, and all 3 tubes got yeast or catalase. So you had enzyme and substrate in all tubes. The difference was the temperature. And so you measured how much gas filled this balloon, right, the circumference of the balloon. And the amount of gas helped you to see how much reaction was actually occurring. And so probably what you saw is that you had an increase in your reaction rate as the temperature went up. So you probably got the lowest in the refrigerator, and then you had more reaction at the room temperature, and you had the maximum amount of reaction at 85 degrees. Now, at some point, the temperature would be so high that the enzyme would become denatured, and then you would see no reaction occurring. But in this case, 85 was probably a very good temperature for this enzyme to be carrying out its reaction.